So you know when we're talking about a muscle, say your bicep muscle, we're actually talking about bundles of muscle fascicles, and each of those muscle fascicles are wrapped in connective tissue, and then each fascicle has within it individual units that are muscle fibers. Each muscle fiber is actually a muscle cell, but it's kind of a funky muscle cell in that muscle cells, when they first are mitosed, they actually fuse together, many of them, up to 50 or more, so that one muscle fiber isn't really a single cell anymore. It's got a whole bunch of nuclei. So when you look at the fiber itself, if you look at the outside wrapping of the fiber, it's actually got nuclei out on the edge. You can see them kind of actually pumping out the side of the nuclei, out the side of the cell. They're wrapped in what is a cell membrane. But in fact, on muscle cells, you have to call the cell membrane a sarcolemma. And in fact, in muscle fibers, instead of calling the material inside the cell cytoplasm, you call it sarcoplasm. The cell membranes of muscle fibers are interesting in that they not only cover the outside wrapping of the cell, you can actually see on the fiber there's these little holes. Those holes are actually places where the cell membrane dives deep into the cell. So it's continuous, that you have membrane on the outside and then it almost goes down into this deep well. That allows, when the membrane depolarizes, the depolarization isn't just on the outside, but it goes down deep into the cell as well. Muscle fibers also have unusual endoplasmic reticulum. The endoplasmic reticulum of a muscle fiber, of course, is not called endoplasmic reticulum, it's called sarcoplasmic reticulum. And it's modified such that they're actually containing calcium, so they're large sacs of calcium. And they are found adjacent to, you actually kind of call it a triad. So where the sarcolemma goes deep into the cell, it's called a T-tubule. And then around the T-tubule, what you see in blue are the cisternae of the sarcoplasmic reticulum, the sacs of calcium. The large portion of the muscle fiber are actually made of proteins that are the contractile proteins of muscle fibers. And you see them here as large bundles called myofibrils. Those myofibrils are made of important proteins that have functions in allowing the muscle fiber to contract. Two real proteins make up that contractile apparatus. The first is a molecule called myosin. Myosin has this interesting structure where it's got the long tail and then it's got the head unit on it. Two myosin polypeptide chains actually wrap around each other, and then hundreds of those myosin molecules get together in an arrangement where every one of these little heads you see is the head of a myosin thick filament, and they arrange themselves almost like a bunch of golf clubs end to end, so the heads are all sticking up sort of on the ends, and the middle part there is bare. So this is called the thick filament, and then the next protein is an actin protein. It actually looks more like a string of pearls. Each protein forms one of these round balls, and the round balls form this long chain that is the actin thin filament. The thick filaments and the thin filaments arrange themselves in the sarcomere in a way such that here's the thick myosin filaments, and here around the edges in the green are the actin filaments. Now that's a 2D image. Imagine the myosin has actins all the way around it. And you notice how they're lined up such that the actins barely overlap with the myosin just on the ends. this would be a relaxed sarcomere. They also have one other type of protein. The myosin-thick filaments have through their core and coming out the end an elastic filament. So right here on the end is the elastic filament. This allows, when the sarcomere contracts, it allows it to relax back. The elastic just pulls them back when they're not contracting. Much like if you take a rubber band, you stretch it out. When you're not putting the stretch force on it and you just let it go, it'll relax back to where it was, and that's what those elastic filaments do. In addition to the contractile proteins, we've talked about the myosin-thick filaments and the actin-thin filaments. There are two other really important proteins that regulate when the myosin and the actin can interact. The first one is called tropomyosin. Tropomyosin are these long strands, and those long strands actually lay themselves out along the actin molecule, blocking all of the binding sites for the myosin. So when tropomyosin is laid out like that, it blocks all the binding sites for the myosin on the actin molecule. They can't bind. You see one other little protein, the troponin little protein, is actually sitting on the tropomyosin. The troponin has a binding site for calcium. So when the cell is activated, calcium gets released from the sarcoplasmic reticulum. The calcium molecules come, bind to the troponin, and when the troponin binds calcium, it has a three-dimensional change that kind of rolls the tropomyosin off the active sites of the actin, so then myosin can bind to those sites. So you can see here the overlap of the myosin-thick filaments with the actin string of bead thin filaments, and when it's fully contracted, the actin and the myosin have bound, and the actin actually kind of crawls its way up the myosin-thick filament. This is showing you the sarcomere. On the top is the sarcomere relaxed, where there's very little overlap between the actin and the myosin, whereas contracted, those actins have pulled their way up the myosin and they're overlapping much more. An important point is that the myosin-thick filaments have not gotten shorter themselves, and the actin-thin filaments have not gotten shorter either. What gets shorter is the entire sarcomere just by the actin and myosin overlapping more. So the more they overlap, that shortens that distance. And when all of the sarcomeres are contracting together, that's enough to cause the contraction or shortening of the entire muscle.